Welcome to our very first lecture on non-chordates. Today, we are going to shrink down and explore a tiny drop of pond water. Inside, we will discover an entire bustling city of microscopic creatures called Protista. These fascinating organisms are the ultimate survivalists, packing all the functions of a complex living body into a single room. And understanding them is the first step to mastering zoology. Now that we know what we are looking for, let us define what a protist actually is. Protists are predominantly single-celled eukaryotes. The word eukaryotic is crucial here. It means their cells have a true membrane-bound nucleus that safely houses their genetic material. Because they are so incredibly diverse, biologists often describe them by what they are not. They are eukaryotes that simply do not fit into the plant, animal, or fungi kingdoms. Despite being just one cell, they have tiny internal organs called organelles to carry out life processes. Before we discuss how they are classified, let us look at what they actually look like inside. Here, you are seeing a schematic diagram of a typical plant-like protist. Notice the long, whip-like structure on the left? This is the flagellum, which it uses to swim through the water. Inside, the large central circle is the true nucleus, the defining feature of all eukaryotes. The green ovals scattered around are chloroplasts, allowing it to produce oxygen and food from sunlight. Finally, the star-shaped contractile vacuole helps pump out excess water. This diagram beautifully illustrates how they pack complex organs into a single cell. Now that we have seen their complex anatomy, how do we organize them? According to traditional classifications, like those outlined by Barnes, protists are broadly grouped based on how they move and how they eat. We have the animal-like protozoans, which zoom around hunting for food. Then we have the plant-like algae, which soak up sunlight to make their own food. Finally, we have the fungus-like slime molds. This simple grouping helps us make sense of their massive variety. You might wonder why we should care about creatures we cannot even see. The truth is, without protists, life on Earth would collapse. Plant-like protists floating in our oceans produce nearly half of the oxygen we breathe every single day. They also feed everything from tiny shrimp to massive blue whales. But there is a darker side. Some protists are formidable parasites, causing devastating human diseases like malaria and sleeping sickness. Understanding them is essential for developing life-saving medicines. Knowing how important they are, we must ask, why are they almost always microscopic? To understand this, we have to look at the physics of their survival. Because they lack a heart or blood vessels, protists rely entirely on simple diffusion to move oxygen and food across their cell membrane. The efficiency of this diffusion depends heavily on their surface area to volume ratio. If a cell gets too large, its internal volume grows much faster than its outer surface area, meaning it will literally starve or suffocate. This physical limitation brings us to a crucial mathematical relationship. We can model a simple spherical protist to see exactly how this works. The formula for the surface area to volume ratio, denoted by R, simplifies beautifully to 3 divided by R, where R is the radius of the cell. Conceptually, this tells us how efficiently the protist can exchange materials with its environment. Notice that the ratio is inversely proportional to the radius. This means as the radius increases, the ratio dramatically decreases, making survival much harder. Before we move on, let us see this with a concrete example. Imagine we are comparing two spherical protists in a pond. The first is very small, with a radius of exactly 1 micrometer. We want to find its surface area to volume ratio. We choose our simplified formula, R equals 3 divided by R. By substituting the value 1 into our equation, we find that the small protist has a ratio of exactly 3. This is a relatively high number, meaning it will easily absorb enough oxygen to keep its internal processes running smoothly. Now let us look at the second protist, which has grown unusually large, with a radius of 3 micrometers. We use the exact same formula to see how size impacts its biological efficiency. Substituting 3 for the radius, the math shows us that the ratio drops significantly to just 1. Comparing the two, the smaller protist is 3 times more efficient at absorbing nutrients. This large protist will struggle to pull in enough oxygen to support its massive internal volume. Now that we have used this formula, let us understand where it comes from. We're setting out to prove how the complex geometry of a sphere reduces to that simple fraction. We start with the basic definition. The ratio R equals total surface area divided by internal volume. For a sphere, the surface area is 4 pi r squared, and the volume is 4/3 pi r cubed. When we substitute these in, we can cancel out the 4 pi and the r squared terms. The 3 from the denominator's fraction flips up, leaving us with exactly 3 over r. This mathematical reality perfectly explains why protists must remain microscopic. So we have proven why they stay small. This brings us to how they multiply. Protists reproduce very quickly through a process called binary fission. This is a simple but highly effective method where one parent cell splits into 2 identical daughter cells. Because every cell divides, the population doubles with every generation. This rapid exponential growth is exactly why protist-caused diseases like malaria can overwhelm a patient's body so quickly if left untreated. To quantify this rapid growth, we first need to know how many times the cells divide. Conceptually, if we know the total time that has passed and how long one division takes, we can find the number of cycles. The formula is N equals T total divided by T cycle. Here, N is the number of division cycles. The variable T total represents the total time elapsed, and T cycle is the time it takes for just one division. If the total time increases, the number of cycles increases proportionally. This formula we just saw leads us to an important question. How many protists do we end up with? We use the exponential growth equation. The formula is n sub t equals n0 multiplied by 2 raised to the power of n. We use the base 2 because the population doubles every single cycle. N sub t is the final population size we want to find, n0 is our starting population, and n is the number of cycles we just learned how to calculate. As n increases, the final population explodes exponentially. Let us apply both of these formulas to a real scenario. Suppose a single amoeba enters a puddle and divides once every 4 hours. We want to find the population after 24 hours. First, we calculate the division cycles by dividing the total time, 24 hours, by the cycle time, 4 hours. This gives us exactly 6 cycles. Next, we substitute our starting population of 1 and our 6 cycles into the exponential growth equation. Calculating 2 to the 6th power gives us 64. This practically demonstrates how a single microscopic protist can colonize a new environment with 64 amoeba in just one day. We have covered a lot of ground today. Let us reinforce the most important ideas. First, Kingdom Protista consists of unicellular eukaryotes that act as a crucial bridge between simple bacteria and complex multicellular life. Second, we classify them broadly by how they move and eat. Third, and most importantly, mathematical principles like the surface area to volume ratio strictly dictate that they must remain microscopic to survive. Finally, their ability to reproduce rapidly via binary fission makes them incredibly successful. You have now mastered the foundational characteristics of Protista, an essential step in your zoology journey.